Hi, my name is Susana Cano, and today I'm going to speak about this article called Interleukin 4 induce 1 in M2, like macrophages promote glioma progression and it's a promising target for immunotherapy. So, first of all, it's important for us to speak a little bit about what's glioma. Gliomas are central nervous system tumors that have physiological features similar to normal glial cells. So, for example, astrocytes, oligodendrocytes, and epithelial cells. So, the World Health Organization has this grading system that is based on physiological criteria that are used to classify tumors as 1 or 4 grades. Now, we're going to speak a little bit about interleukin 4 and interleukin 1. Taking into account that in this study it's using interleukin 4 induce 1, so it's a mix of both. The most important thing we need to talk about is where they came from. So we have this beautiful diagram that's going to tell us that from a stem cell, it's going to be differentiated into a monocyte of macrophages, and from that one, we're going to have the interleukin 1. But from the interleukin 4, we're going to have a differentiation into a lymphoid prosecutor, then into a T cell, differentiated into CD4 T cell, and then into a GATA3, and then we're gonna have interleukin 4. So, what are like the cells that they're gonna target? So, interleukin 1, all type of cells. Interleukin 4, specifically for TB and NK cells, monocyte macrophages, neutrophils, eosinophils, endothelial cells, and fibroblasts. And the biological activity they're gonna have is the interleukin 1 upregulates adhesion, neutrophil and macrophage emigration. They can mimic shock and fever, and hepatic acute phase protein production, and one lastly that's really important is that they stimulate hematopoiesis. Interleukin 4, on the other hand, can be used for T-cell differentiation and proliferation. B cells, immunoglobulin class, can switch into immunoglobulin G type 1. The general objective is identify interleukin 4 induce 1 in M2 macrophages type 2, like macrophages promote glioma progression and as a promising target for immunotherapy. About the methods. So we're gonna have like two slices of them. So first of all, we have the Western blot and the PCR. These specific two are gonna be working as team, so they complement each other's information to know how the cells are working. So first of all, we have the Western blot. Western blot works to identify proteins in a sample. Basically, what they do is that protein antigens are separated by size in a gel and electric field, and the antigens are then transferred from the gel onto a filter paper. So those antibodies are then added. PCR. It's important to know that this is a quantitative real-time PCR because the normal one is a qualitative method. So basically, as a quantitative method, it's gonna be measuring the amount of RNA in a tissue to assess the level of the gene expression. Basically, make copies of the DNA. They're both working for the same objective. So it's basically to analyze interleukin 4 induce 1 in relation with antibodies. So we're gonna have targets for the different types of antibody that are connected with different types of macrophages. They are important because they allow us to know the patterns of the interleukin 4 induce 1 and its expression between the role across 30 gene tumor types. Compares the differences in both mRNAs. So at the final, we're gonna have silence interleukin 4 induce 1, and it's important because with these studies, it can allow us to know how it behaves in connection with the CD206 and CD163, and how it has no effect for the CD86 that is basically related for the macrophages type 1 as methods that are immunochemistry and immunofluorescence. So they work as a different type of methods, but they work for the same purposes. So first of all, immunochemistry uses labeled antibodies to localize cell and tissue antigens, and its most specific is a chemical method. So now to speak about immunofluorescence, we're gonna have that this one is used to detect determined antigens in a cell or in a sample. The antibodies have an other special staining patterns and they unite with the antigen of the cell. It's used for determination of proteins, drugs, and hormones. But speaking about the article, we're gonna know that these two are gonna be used for, first of all, to represent the quantification of migration and invasion by the cultivation of glioma cells with M2 macrophages to know basically how it's working, the glioma, and the connection between the M2 macrophages. Also, to check if the immunofluorescence differentiate interleukin 4 induce 1 in macrophages and glioma cells. Also, the relation between the CD206 and the interleukin 4 induce 1. And lastly, the impact of the silenced interleukin 4 induce 1 and the proliferation of two types of glioma cells results. So we have, first one, it's immunofluorescence. That is like a saying that it's going to be for reference. The CD206 that it's related for type 2 macrophages and interleukin 4 induce 1. So basically, we're gonna see it's like these two are connected. So everything is superposed between them. Basically, it's not like a really important or significant graphic, but here we have a Western blot where we can check different types of antibody and determine that's basically the reference and how they are connected vertically with the different types of macrophages. One important thing that we have to know is that the first one, the THP1, it's a kind of monocyte cell line. So it's macrophages before they differentiate. So more important things in this graphic are gonna be the increase of CD204 in the presence of macrophages 1 and 2. Basically tell us which macrophages are connected with which type of antibodies. And for these graphics, we're gonna have the relative protein expression and the relative mRNA expression. So it's important to know that these two are gonna have like similar correlation between the results because mRNA in the future eventually it's gonna become a protein. So they must have some relations, but they can be different. So in the graphic, it's basically what it's gonna be already in the Western blot. So the most important things are the difference between the CD204 and the CD163. We're gonna be checking the different type of macrophages in connection with the interleukin 4 induce 1. But surprisingly, we're not seeing anything in the glioma cells. So this is kind of connecting with the conclusion of the article. We have the connection of these two types of staining and the connection between the macrophages and the glioma cells. Now we're going to speak about the results in the figure 7. These results are not significant because, as you can see in this graphic, they are saying it's not significant because the results are almost the same. So this graphic basically it's a connection between this new type of staining and the connection between these glioma cells and the silence interleukin 1. Then we have the relation between the antibodies that we already speak on, interleukin 4 induced 1, that is this line, and the relation between the M2 macrophages and MO macrophages having the silence interleukin 4 induced 1 and then not silence. Okay? So the most significant difference is that for the silence ones, we're gonna have a decreasement the booster blood. Well, speaking for this graphic B, it's basically what we are kind of seeing here, like the connection between the silence or no macrophages and with the different color stainings. This one is the DAPI and this one is the CD206. So this one and this one are gonna have a less amount of staining. Discussion we have neither that about the bad prognosis we have in glioma, Afshan and PAP. Clean means tumor-associated macrophages. A connection with the proliferation and metastasis of tumor cells. And lastly, we have Morrissey. The TMAs are also unknown factors associated in the prognosis of glioma. So it's basically a connection between these two ones. As a conclusion, we have that molecular biology allows us to develop new techniques for developing innovations by going to the microscopic causes and principles. We're trying to say that sometimes we don't need these big and super crazy ideas to have solutions for our daily issues. We can go to the main causes to work by that. And this is also going to help us in different things because if you have a principle as a small thing, it can be divided to multiple things. So the second conclusion is that this article is key for new common discoveries in glioma pathology, as it allows a more specific approach where more studies can be made. As I was saying, having this new knowledge about interleukin 4 induced 1 can allow new scientists to work with this information to use it as a targeting for diagnosis or for human treatment. So we are focusing on this interleukin.